Hello everyone, welcome to Last Night Dentistry. Just imagine if I told you that the hardest tissue in the human body is actually built by a single layer of cells. Even more surprising, once these cells disappear, your body can never make enamel again. That's exactly why understanding amelogenesis is so important. It's one of the earliest topics in oral biology, but it becomes your foundation for pathology, pediatric dentistry, operative dentistry, and almost every licensing exam like ORE, ADC, and INBDE. By the end of this video, you will understand the entire process in under 10 minutes, and I promise you will never have to memorize this topic again. Starting where, what is amelogenesis? Let's start with the big picture. Amelogenesis simply means enamel formation. One liner answer is enamel formation. The cells responsible are called ameloblasts. Think of an ameloblast as a highly specialized construction worker. Its only job is to build enamel. The process happens in two major steps. First, the cell lay down the enamel matrix. At this point, enamel is soft and only about 30% mineralized. Then comes the second step. Instead of adding more enamel, the cells remove protein and water while packing in minerals. That's how enamel becomes over 96% mineralized and earns its title as the hardest tissue in the human body. Keep this idea in your mind because everything else in this chapter revolves around these two stages. Now let's see the whole journey before we study each stage. Everything begins with the inner enamel epithelium. These cells first prepare themselves. This is called the pre-secretory stage. Next, they actively produce enamel during the secretory stage. Once the full thickness of enamel is formed, they enter the maturation stage, where enamel becomes harder, and finally they protect the enamel until the tooth erupts. If you remember just the sequence, you will already answer many Viva questions correctly. So, moving to the pre-secretory stage. The stage is all about preparation. Think of it like a chef getting the kitchen ready before cooking. So the cells become taller, their nucleus shifts to one side, the rough endoplasmic reticulum and Golgi apparatus become much larger because the cell is preparing to manufacture large amount of proteins. The basal lamina also disappears, allowing future enamel to come into direct contact with the dentine. One important point students often miss is that no real enamel is being deposited yet. The cells are simply transforming into fully functional ameloblasts. So don't think of the stage as enamel formation. Think of it as enamel preparation stage. Okay. Now moving to secretory stage. This is the star of the entire chapter. Now the ameloblasts began doing the job they were designed for. Inside the cell, enamel proteins are produced in the rough endoplasmic reticulum, modified inside the Golgi apparatus, packed into secretory granules, and finally released onto the tooth surface. As soon as these proteins are secreted, enamels become forming. Notice something interesting. Enamel does not wait to mineralize later. Mineralization actually starts immediately. However, it's still only about 30% mineralized, which means it's far from becoming mature enamel. Now, moving on to the Toombs process. If there is one structure every dental student should remember from this chapter, it is the Toombs process. This small projection develops at the end of the ameloblast and acts like a mold that shapes enamel. It creates enamel rods and interrod enamel, giving enamel its unique microscope architecture. Here's a favorite Viva question: why is the very first layer of enamel rodless? Because tomb process has not fully developed yet. Later, when Toombs process disappears at the end of the secretion, the last layer also becomes rodless. So remember the simple rule: no Toombs process equals to no enamel rods. Moving on to the enamel proteins. Students often panic when they see so many protein names. Let's simplify them into three characteristics. First, Amelogenin. Amelogenin, this is the main protein. It makes up about 90% of the enamel matrix and controls crystal growth. Second, amyloplastin. Its main job is helping ameloblasts stay attached to the enamel surface. Third, enamelin. Think of it as the protein that helps crystals start forming and grow properly. You don't need to memorize every molecular detail, just remember the job of each protein. Moving on to the maturation stage. Now comes the transformation. No new enamel is added, instead, the enamel that already exists becomes stronger. Imagine like building a brick wall, the wall is already standing. Now you are filling every tiny gap with cement. That's exactly what's happening here. Protein leaves, water leaves, and minerals enter. The crystals become wider and thicker, and mineral content rises from about 30% to over 96%. This is why the maturation stage is essential for producing strong enamel. Students usually find these names confusing because they are actually very logical. Ruffle-ended ameloblasts mainly add minerals into the enamel, while smooth-ended ameloblasts mainly help remove protein and water. Think of it this way: one worker delivers new building material, the other cleans away the waste. These two forms continuously alternate until enamel reaches the full maturity. Now, another very common exam question is about these two enzymes. MMP20 works early during the secretory stage. It trims newly secreted enamel protein so enamel can develop normally. KLK4 works later during maturation. Its job is much bigger. It breaks down the remaining protein so minerals can completely fill the space. An easy way to remember this is MMP20 starts the cleanup, KLK4 finishes it. Moving on to the once enamel is fully mature, the ameloblasts don't simply disappear. They become something called reduced enamel epithelium. Its main job is protecting the enamel surface until eruption. Later part of this tissue contributes to the formation of junctional epithelium around the tooth. This is why oral biology connects directly with periodontology. Now we got everything we have learned finally explains why enamel defects occur. If something goes wrong during the secretory stage, less enamel is formed, resulting in enamel hypoplasia. If the problem occurs during maturation, enamel forms but does not mineralize properly, leading to hypomineralized enamel. And when these developmental problems are inherited genetically, we call the condition amelogenesis imperfecta. Later on, we will discuss the diseases as well. Notice something important. Once enamel is damaged during development, the body cannot remake it because ameloblasts disappear after tooth eruption. Here's the rapid revision before you leave. Let's quickly test ourselves. Which cells form enamel? Which states form enamel rods? Why is the first enamel layer rodless? Which enzyme work during maturation? If you answer these without looking back, you have already mastered the highest yield concept from this chapter. If this explanation helped you finally understand amelogenesis instead of memorizing it, subscribe to Last Night Dentistry because we will cover the rest of our bio in the same simple exam focused way. I will see you in the next one. Thank you for subscribing.